Biochemical tests are laboratory procedures to differentiate various bacterial species. The first one is catalase test. This is used to detect the enzyme catalase. This enzyme is responsible for protecting the bacteria from hydrogen peroxide accumulation. This can occur during aerobic metabolism. If hydrogen peroxide accumulates, it becomes toxic to the organism, and catalase brings hydrogen peroxide into water and oxygen. The method: on the surface of the clean test or a slide, mix few colonies with a drop of hydrogen peroxide, and the production of effervescence or bubbles indicate the positive test. This test is done on a slide or on a test tube, as I told you already. It is an essential test to differentiate between the genus of Staphylococcus and Streptococcus. Staphylococcus gives a positive test result and Streptococcus gives a negative test result. Okay, I think you are clear about this. So this is the representation. So these are the effervescence which are found. The first one describes the tube test, which is on the right, and the far left represents the slide test. Next is coagulase. This is used to identify microorganisms that can produce the coagulase enzyme, which is helpful in the identification of Staphylococcus aureus from other coagulase negative staphylococci. Okay, so what is the new name of Staphylococcus aureus? Other cons? Can you say what is the other name of cons? It is SOSA. It is Staphylococcus other than Staphylococcus aureus. Okay, this is a new nomenclature. So this coagulase test and catalase test helps in differentiation of Staphylococcus aureus. Here Staphylococcus aureus is coagulase positive and catalase positive. Okay, so here you can also see that coagulase is also one of the most important virulence factors found in Staphylococcus aureus. During the reaction phase, the coagulase enzyme will coagulate the blood plasma, and this test is carried by combining blood plasma with the bacterial colony. So the bacteria generates a coagulase enzyme which causes the blood plasma to coagulate, indicating a positive. The method: so there are two types of coagulase. The first one is bound coagulase and second one is free coagulase. Bound coagulase enzyme is done by slide test. So here the homogeneous suspension of the test or bishop is made into a drop and saline is added to it. So both are mixed together, after which you take one drop of undiluted human or rabbit plasma and mix it together. Later what you do is you take a 10x or a low power microscope or a magnification lens and observe the clumps for positive testing. The second one is tube coagulase method. Here you add 5 to 10 drops of Staph aureus colony mixed with normal saline, then take 1 ml of human plasma or rabbit plasma. So the relation should be 1:6 in a sterile saline, and incubation should be done for around 4 hours at 37 degrees, and the clot should be examined at 2 hours and 4 hours. If not found after 4 hours, you should wait for 24 hours and then identify whether clot is present or not. Okay, so this is how the coagulase test represents. The first one is positive test result and the second one is negative. Next is oxidase test. Oxidase test is used to identify if the bacteria is able to produce cytochrome C enzyme or not. Okay. The procedure: take a filter paper soaked with substrate tetraethyl phenylene diamine dihydrochloride. So this is the name of the oxidase reagent. Okay, so we can simply say that take the oxidase reagent, moisten the paper with the sterile distilled water, pick a colony to be tested with a wooden or platinum rope. Do not use any other material like iron because it causes false positive reaction. Then observe the inoculated area for color change to deep purple or blue in around 10 to 30 seconds. Okay, more than 30 seconds is not valid. Next is indole test. Indole test is used to determine the ability of a bacteria to degrade tryptophan by the enzyme tryptophanase. So tryptophan is degraded by using tryptophanase containing bacteria. The test is performed by inoculating peptone water containing tryptophan with the test bacteria and incubating at 37 degrees for around 16 to 24 hours. After incubation, the few drops of coax reagent is added. A red ring in superficial layer of the medium indicates a positive result. So examples of bacteria producing indole positive are E. coli. Negative reactions are yellow in color, and organism which is causing such reactions are Klebsiella, etc. The urease test. The urease test is the ability to identify microorganisms that hydrolyze urea and use urease enzyme. The procedure is treat a surface of urea agar slant with a portion of a well isolated colony of organism suspected to contain this urease enzyme and incubate it around 48 hours to 7 days at 37 degrees. Examine for the development of pink color. So organisms which are positive for urease reaction are protease species, example Proteus mirabilis and vulgaris, and Escherichia coli are negative for urease test. Next are IMIC test. So IMIC test consists a series of tests which include the following reactions. The first one is indole protection, second is methyl red test, Voss-Prescar, and citrate nucleation test. These tests are a group of biochemical tests that help in identification and differentiation between entry gram-negative bacilli of the species Enterobacteriaceae. Thank you. I hope this lecture would be helpful for you and keep supporting us for such useful videos. Thank you.